Welcome to our podcast on connective tissue. This is for our connective tissue lab, and we're going to be using what I again consider the red line or the label type of illustrations that we've seen on the internet before. And we're starting with basically a mesentery that we might remove from some part of the body. This might be from the mesentery holding on to the small intestine, connecting it to the body. I'm not exactly sure where this comes from, but as we look at this and zoom in more closely to the tissues, we're going to see blood vessels in the tissue. We see some nerves, capillary networks. But as we look at higher and higher magnification, we're going to see more and more detail and be able to visualize more and more of the cells and the molecular components that we see within connective tissue. Much of what we see in these circular structures are going to be adipocytes. You find a lot of tissue associated with mesentery. Now, this is not a magnification that I would expect you to visualize adipocytes, but as we go through and see these, you will certainly be able to suspect, even at this magnification, we are looking at fat cells. Now, this is a very high magnification, and as we look at this image, we see many fibers, and then we can see, even though they're out of the plane of section, a lot of nuclei. We can even see a cell over here, the cytoplasm nucleus in this point. And what we're looking at, these very thin, dark filaments that are actually in the plane of section, these are going to be our elastic fibers. That's what we're looking at. And if you can sort of imagine and change your focus, so behind the elastic fibers, out of the plane of section somewhat, these much larger diametered fibers that we see and running in parallel right here, these are going to be our collagen fibers. So we have fibrous components, elastic, collagen, a lot of space, cells scattered throughout here. This would be our loose or areolar connective tissue. So there are elastic fibers. Now this arrow appears to be pointing here, but understand we're pointing to the fibers behind and sort of out of the plane of section. You can see it running there. This is a nice close-up view of our adipocytes. So we have our triglyceride material inside, very thin rim of cytoplasm, the nucleus very peripheral and oval shaped and flattened. So this is our typical white fat or unilocular adipose tissue. We have some capillaries running through here. We can see red blood cells in the single file. We can also see our donut-shaped red blood cells running through this capillary at this point. So we have white fat, adipose tissue, some blood vessels running through here. So a nice example of that type of connective tissue cell. Now we move into looking at more tissue. And again, we're not supposed to be identifying anything other than epithelia and connective tissue at this point. But since the connective tissue interconnects different types of tissues, we're going to see more things than we're really familiar with at this point. What we're looking at in the bottom part of this image, this is going to be muscle. And we have connective tissue in between the muscle and these other layers. Some of this connective tissue is going to be of the dense variety, and some is going to be of the loose variety, and we'll have dense regular and dense irregular. And we'll go up in higher magnification as we look at this. But as we look at this illustration, we can see our dense regular connective tissue. In this case, this is going to be our tendon. Here we have our skeletal muscle. This is where we can see our loose or our areolar connective tissue, a lot of space in this region. Then somewhat of the capsule that surrounds this dense regular is our dense irregular. We can see it below and we can see it above. Let's just go back. Right there we can see how dense that connective tissue is. Very little, if anything, in white space, and these are probably artifact, again, of our preservation of our tendon. All this white space, kind of the marbled look. Think about a steak, and that's how you'll be able to identify some of these sections of skeletal muscle. A lot of white space in here in our loose tissue, more dense, but as we begin to look at a higher and higher magnification, we're going to be able to see how we do have more space. The fibers are not as parallel as we see in our dense connective tissue. And that, of course, is going to be our dense collagenous connective tissue in the tendon. Higher magnification of that inset, here's our tendon. are very elongated nuclei of these cells in our tendon. And do you remember what those cells are going to be? These are our fibroblasts. And as we get toward the periphery, this is our dense irregular. You see the white spaces, and it appears to have a pattern to it. These are mostly parallel, but again, you see more of that white space. It's not as compacted. Our dense regular connective tissue is ligament and tendon. It's going to look like this. Any space, even if the fibers are somewhat more parallel, then we're going to have dense irregular connective tissue. A lot of blood vessels interspersed in that as well. Here's another view. This is a cross-section of muscle and tendon. We can see the marbled skeletal muscle. Some of these muscle cells are fascicles of muscle, and we'll deal with more of that when we get to that muscle section. But here we have our tendon that's going to be cut in cross-section. And again, we'll look at that in higher magnification. So now we've got our cross-section of muscle, and we'll be able to understand all the things we need to see to identify that as muscle. But in our tendon, we no longer see those nice elongated nuclei because we're cutting them in cross-section. They're just little spots. But we see no space whatsoever. We're looking at the cut ends of parallel arranged collagen fibers. And so when you just have this homogeneous field, so sparsely consisting of cells, that's one way that you could determine that we're looking at a cross-section of dense, regular, collagenous tissue, and that will be tendon. We pretty much can extrapolate that we're looking at tendon because of the close association with muscle. Ligament would look exactly the same, it's just the interconnection of bone to bone. Now we're shifting gears a little bit as we move into other types of connective tissue. Here I will go ahead and let you know we are looking in our respiratory system. We're going to have some muscle. This is going to be smooth muscle down here. We've got our cartilage. We have our connective tissue that underlies our mucosa and our epithelium. Now, when we talk about organ histology, and I'm already using terms we're not familiar with, but in organ histology, such as respiratory tract, digestive tract, our epithelium we're going to call the mucosa, and the connective tissue underlying the mucosa is going to be the lamina propria. Those are some terms that we're using, but we're just going to look at those connective tissue spaces as we move through this. So there's the lamina propria that you see, and that's where we're going to be looking at our connective tissue. All right, a little bit higher magnification looking in here. Again, we're going to be focusing on the lamina propria. Certainly, our cartilage is going to be a form of specialized connective tissue. And in our lamina propria, we're either going to have the dense irregular connective tissue, or sometimes in our lamina propria, we might actually see loose or areolar connective tissue, depending on how many spaces and how many cells we might have in a particular section. This looks to be dense irregular in this particular slide. Now we're going to be looking at cells again. We are in a field, and it's difficult to tell at this high of a magnification if we're looking in loose or if we're dense irregular. There are quite a few fibers in here, so my gut reaction is we're going to go with dense irregular. So we're like in lamina propria as we saw in the previous image. I don't exactly know where we're looking. But as we look at the cells in this field, we've got these really dark round staining cells, probably a lymphocyte, but these are the ones that we really want to focus on. Not quite as blue staining, large oval shaped nucleus. I'll give you a hint, you can imagine sort of an indentation in the nucleus, but what I really want you to see are these bubbles, this soap bubble-like appearance. These are the vesicles, maybe phagocytic vesicles. These are macrophages, resident macrophages in our connective tissue, and that's how they're going to look in a high magnification. Once again, we have a lot of cells to deal with here, possibly have a fibroblastic nucleus that's present, but now you can see all of the bubbles in the cytoplasm of this macrophage, quite a few macrophages present here, and we have some leukocytes, white blood cells, that are going to be present in this field. You can see the multiple lobes of the nuclei, and that's how we identify those. Not going to spend a lot of time talking about them right now, simply because we'll get to them when we talk about blood. Once again, more cells in our connective tissue. Nice examples of fibroblastic cells, elongated, tapered nucleus. We've got some blood vessels in here. And then we have these cells that stain with these dark red granules. This is an eosinophil. It looks binucleated, but eosinophils have bilobe nuclei. One nucleus, but in this plane of section and its cut, it looks to be two. Now, this is a great example of a plasma cell in its characteristic clock face nucleus. You can see the dark U chromatin, and it almost resembles the numbers on a clock face, and a very prominent nucleolus, which is going to be right in the middle. You also have this light supranuclear region that you're going to have in the plasma cell. Makes this sort of a light region around the nucleus. So that's going to be a nice example of a plasma cell that we have in our connective tissue. Again, we've got more connective tissue. We can see our epithelium on the right in this location. This is going to be our pseudostratified columnar epithelium, and we do have cilia on the surface. You can see it very faintly. I wish we had a better, more contrasty view here, but we've got the cilia on the surface. We see some blood vessels with the simple squamous epithelium. We've got our fibroblast in here, and possibly another plasma cell. You don't see quite the intact nucleus, but again, the U chromatin's making sort of the numbers on the clock face, the nucleolus in the middle, and then this lighter supranuclear Golgi region. So I believe we're looking at another plasma cell. More cells once again. This is really nice because you can see the red blood cells inside this blood vessel. We've got our fibroblast. Here we have our epithelium, pseudostratified columnar. With this stain, the goblet cells and the mucin granules are staining dark. And there you can see the ciliated border on these. That dark line, that's going to be our basal body layer. Here we have another plasma cell with a clock face nucleus. And here we have a really, really dark cell, large granules, and the granules practically obscure the nucleus, these dark blue staining granules. This is our, if it was in our circulation, we'd refer to this as a basophil, but out in our tissues, that's our mast cell or tissue basophil. Now, in this particular field, we're still in our respiratory epithelium, pseudostratified columnar epithelium, ciliated. And in this connective tissue region, there are a ton of lymphocytes and plasma cells. We are going to discuss these in our lecture podcast. This is going to be transient lymphatic accumulations. They're called mucosa-associated lymphatic tissue, or MALT. And we're going to see these in respiratory tract. We have a lot of these in our digestive tract. Much of what you're seeing in here is going to be lymphocyte tissue. You'll have fibroblasts as well, but this is just a whole conglomeration, mostly of lymphocytes, plasma cells. You see there, they're labeling some of the macrophages. Those are so challenging to point out. They're larger cells. Now that you point them out and go back and forth, you can somewhat pick them out. I would wager we possibly have another macrophage right in here as well. But at this magnification, I would not ask you to identify any particular individual cell in these. Once again, very high magnification. This is really cool, looking at our capillary single endothelial cell. So I guess you could honestly classify this as a simple squamous epithelium. That's going to be kind of hard to do since it's one cell, but you can see that one red blood cell is barely squeezing through that luminal diameter. There we have another one out of the plane of section. Here we have another one of those cells. Looks elongated. You might first think, ooh, it's elongated, it's a fibroblast, but the dark granules, blue staining, it's another mast cell. Still working with respiratory epithelium. Lymphocytic tissue is also going to have a lot of our leukocytes. And these red granules, again, bilobed nuclei, all of these are our eosinophils. The color of the granules sometimes might be harder to see than others. And for me, if I can see the lobes of the nuclei with these different stained granules, that helps me know that we're looking at eosinophils. These are nice red, so that contrast really helps to bring that out. And another image, slightly different contrasting picture to help us be able to look at these. You can see our respiratory mucosa, certainly, our fibroblasts running throughout this tissue. So this is our dense, irregular connective tissue just beneath the epithelium. All right, changing gears a little bit. We've been looking at a lot of our collagenous connective tissue. Now we're going to be looking at other types. We've got a lot of fat here, and this is going to be a place where we're going to distinguish the unilocular from the multilocular adipose tissue. We'll be able to see these tied together in the same place. So looking at higher magnification, we're looking for the droplets. And in this case, we have our nice signet ring appearance, one large droplet. And I know that you see the granular look. I think that's just an artifact of the preservation or the staining. This is one large lipid droplet. We can see the nucleus really squeezed to the periphery. This is unilocular adipose tissue. All of these, in fact, are unilocular. But these are our multilocular. You see how the same appearance to these, but the vesicles are much smaller. And there's many more of them. So this is our brown fat, multilocular. This is our white fat, unilocular adipocyte. Now, in this particular illustration, what's happened is we have taken tissue and we have digested away the cells and the other softer tissue. We're looking at the fibrous skeleton, the fibrous extracellular skeleton of a particular organ. So these are going to be our reticular fibers, type 1 and type 3 collagen, just to give you an idea of the scaffolding on which cells hold. This is going to be a blood vessel, and you can see the red blood cells inside of this that still remain. Now we're at a low magnification. Certainly, hopefully, from what we have been able to see in our virtual slides that we did, you will be able to even recognize this as our stratified squamous keratinized epithelium, or a part of the skin. And as we look at the integument in skin, we're going to break it down into our epidermis, dermis, and then our hypodermis. This is going to be our superficial fascia layer that has a lot of adipose tissue in it that we're going to have covering our body. And so within this region, we can see a lot of our typical white fat, signet ring appearance. This is our unilocular adipose tissue. Once again, on the left, we have a nice example of our stratified squamous keratinized epithelium. We have our dermis, which is our dense, irregular connective tissue, and then a lot of adipose tissue here on the left. Nice blood vessel. And the black that you see, this is a particular stain that's going to be detecting with black the elastic fibers. So you can see the elastic nature of this layer in our dense irregular connective tissue. And you can even see some of the elastic lamina around this blood vessel. So this is not just collagen. We have a high density of collagen there, but we also have elastic fibers as part of that. Now we're shifting gears just a little bit because we're going to focus on connective tissue cells and the microenvironment around cells from a very high magnification. And these are going to be transmission electron micrographs. And these are always going to be black and white, sort of grayscale, unless they're computerized and pseudo-colored. And we have already seen this one in our lecture for connective tissue. We have a fibroblast, elongated shape, elongated nucleus. We have our fibrils that we see here in longitudinal section, cutting cross section. Do you remember from lecture what these are? Now again, you may be tempted to think that we're looking at striated muscle. You can see some of the striations here, but you have to remember the scale that we're looking at. This is an entire fibroblastic cell. Our muscle is a lot bigger than that. So we're on a very small scale here. And so what we're looking at is type 1 collagen in longitudinal section and collagen cut in cross section as we look at the fibroblast. Once again, we have a nucleus. Do you remember the dark material around the periphery of the nucleus? That's going to be our euchromatin. Heterochromatin is the light color, and we really don't see a nucleolus there. What about these structures, these parallel railroad track structures? The rough endoplasmic reticulum, you can sort of see the rough area there. What about this right here? This is cutting cross section, so it might be a little bit more challenging to see that. That's a mitochondria, and we have some little vesicles that we see budding in there, possibly there, another one there. Now this is one of those insets that we saw just looking below the fibroblast, and here you can see the nice periodicity of our collagens, that regular row. And unlike what we would see in our myofibrils in muscle, we don't see any alternating light and dark bands. You just see these light areas of periodicity between the collagen molecules as they're put together. And here you can see the collagen cutting cross section. Nice look at some endoplasmic reticulum, and you can see the individual ribosomes on the ER there. Okay, we're looking at different cell types here. Now what we're going to be looking at in our connective tissue, we're getting into the more specialized cells, our leukocytes. We can see very specific granules in these. And this cell is going to be larger than what we'd seen before. We're not seeing indented nuclei, but we're seeing these vesicular structures inside, dark in nature, some of them, and some are going to be lighter in color. At the light microscopic level, these are going to appear as soap bubble-ish. And so this is going to be our macrophage. We've got some processes coming off of these cells. We've got just connective tissue in this area. Once again, we've got our euchromatin, the heterochromatin, the lighter color inside. And we will not be calling these histocytes. It's a macrophage in the tissue, and in the circulation, what do we call it? A monocyte. Now, one of the insets, this is our eosinophil, and this is our specific granule, and we'll go into more details about these specific granules in our lecture, but you can see we've got an outer dark area, but then we've got this inner crystalline-like structure. This is going to be the centrum of our specific granules. It's not really critical. You remember that now. We're just pointing that out, and you can remember that as we get down the road. Here we have some of our phagolysosomes, this material that's present inside these vesicular structures of the macrophage. Now this is a cell we'll see quite a bit of in our connective tissue areas, especially our loose or areolar connective tissue, and it's the cell that has the clock-faced nucleus. We can see the euchromatin around the outside in clumps. We can see this larger clumping right in the middle. At the light level, it appears to be a nucleolus. This is actually the nucleolus, this little condensed spot here. But you're going to see that dark euchromatin in the middle. Tons and tons of rough endoplasmic reticulum scattered out here with mitochondria. We've got some expanded Golgi regions in here as well. This is a plasma cell. This is what a plasma cell looks like at the EM level. Here we see some real high magnifications of our ER. You can see the granular look on the endoplasmic reticulum, and here you can see our Golgi apparatus, and we have our transport vesicles coming off of the Golgi here. You can see some more of those Golgi's transport vesicles here. Now the last cell we're going to look at, this is the cell that really can give us a lot of problems, especially as it relates to our allergic reactions. We see a lot of these specific granules. These are much larger, and you see they're more round than we saw with the eosinophil. We're not necessarily going to see that externum and internum structure that we saw, the crystalline structure. These are going to almost obliterate the nucleus at the light level. We won't see that. And you can see we've got a lot of these processes as this cell is in the process of degranulating. This is going to be in the tissues. This is going to be our mast cell. And in our circulation, this is called, what do we call a mast cell in circulation? A basophil.